So for the last couple of years, there's been an explosion of publication and news on CRISPR. So I'm going to give an easy and in-depth discussion on what CRISPR is and why it's such a big deal. So let's start with the discovery. So in 1993, they were comparing genomes of different microbes, and they found that there was a sequence that was repeated among different bacteria. So this is where CRISPR got its name from. So there was an observed cluster of short palindromic repeated sequences evenly spaced throughout the genome. So if we have our bacteria DNA here, notice that they noticed these regions here, black. They were repeated sequences that were palindromic, and palindromic just means it's spelled forward and backwards the same way, so as taco cat is spelled taco cat no matter which way you look at it. And so if we look at nucleotide sequence, it might be like AGGA, where it's spelled AGGA frontwards and backwards. So that was observed in these repeated sequences in black. And then they were flanking these unique sequences that were called spacer sequences, which I'll get into later. And then in 2005, an explosion of research really began when we started to put together what this region, this repeated region was. It was part of an immunity, bacterial immunity. So what is CRISPR? Well, we can look at adaptive and learned immunity to get an idea. So let's compare it to humans' adaptive learned immunity. One of them would be B cells. So a pathogen infects, a B cell recognizes a specific antigen, and it produces antibodies. For bacteria, a virus infects, and the CRISPR system is going to recognize a viral sequence from that virus from a previous exposure, and then it sends a complex to destroy the virus. So the virus DNA. Inside of that DNA right here is a specific sequence that is going to get cut. So this whole thing gets cut, virus is dealt with, and it goes away. Let's get some more background information on CRISPR before we dive in. So it occurs in about 45% of bacteria and 83% of archaea. Now, the study was done in, I think, 2015, so the numbers could change, but the overall idea is that not all bacteria and archaea have this system, but a lot of them do. And the way that they differ is that each bacteria will produce certain proteins to carry on this CRISPR system. So there'll be Cas proteins, CRISPR-associated proteins that differ. And this is what gives us different classes, so class 1 and 2, and the different types within those classes. Mostly, we look at this type 2 system because it's the simplest and easiest one to understand and utilize in the lab. So the general pathway of CRISPR is 3 steps. So there's adaptation, where you get a viral sequence from a virus, this is a spacer, and then there's biogenesis and expression. This is just putting together your CRISPR RNAs and your complex, and then targeting, where you're going to attack the virus that's infecting. Let's look at the first step, adaptation and acquisition. So we're going to focus on the type 2 system. So Cas1 and 2, those are CRISPR-associated proteins, are made by the bacteria. So they produce these proteins. A virus infects its DNA. The proteins attack that DNA. They cut up that viral sequence, and now there's these little fragments of viral sequences inside this bacteria. The Cas proteins help associate this viral sequence into the bacterial double-stranded DNA. So this is now part of the CRISPR array. This is where we have the repeat sequences in black, and then each of these spacer sequences, you might have guessed, are actually from a virus most of the time. And so this is a memory bank of previously encountered viruses. So how many viral sequences can be stored as spacers in this bacterial DNA, and what stops it from growing indefinitely? Because we keep taking this sequence and we're inputting that sequence into the double-stranded DNA so we can recognize it later. What stops it from taking a lot of these viral sequences from multiple viruses and just expanding throughout the genome? Well, first let's look at how big these sequences even are. So the repeat sequences are about 30 base pairs, and the spacer sequences, which are the viral sequences, are around 20 to 80 base pairs, and they can be up to 72. So an example of this is Sopholibus tocodi. So there's 5 CRISPRs uh, regions. So that's upstream. That's going to be the Cas proteins and Cas9, stuff like that. And then there's 458 spacers. So these are the viral sequences, and about one, which make up about 1% of the genome. And it's actually been found that deletion events do occur. So this is called array erosion through spacer deletion. And that's probably what limits the spacer acquisition from just growing indefinitely. They start to degrade, and then new spacers can come in. So let's look at the second step. This is the biogenesis step of the type 2 system. So upstream of the DNA of this CRISPR locus, this is the CRISPR locus, or also called CRISPR array. Just upstream of that are some CRISPR genes. So one of them is going to make a tracer RNA. Another one is going to make a Cas9 protein once it's fully translated. And then we want to take this whole CRISPR array and make a mRNA. So this is a primary mRNA, hasn't been cut up yet. The tracer and Cas9 are going to associate, and tracer is going to help bind it to the mRNA. It is going to bind homologous base pairing between that repeated sequence. This is the repeated sequence with that tracer RNA. So this tracer is going to have homology to that repeated sequences, which are palindromic. But now we need to cut it up more. So because we have this giant long mRNA, we don't want that. We want short complexes of this repeat sequence and a spacer. That's it. So we have RNase 3 in the bacterial cell that binds in, it gets recruited, and then it cuts. So now we have these little fragments of complexes which have a spacer. Remember, the spacer is that viral sequence, and then a repeat sequence. That repeat sequence helped guide this complex there. And each of these is going to be looking for a certain virus because of that spacer sequence has homology to a specific virus that it took. So it took a piece of that virus during that first step, and now it can go ahead and attack it. You can see that this system's pretty easy. It has 4 components. Cas1 and 2 are used in the acquisition step, taking that viral DNA. Cas9 and the tracer are used in biogenesis and targeting. And then the primary transcript is really that giant mRNA that's formed, and it recruits that RNase 3 just from the bacterial that already has it floating around. So let's take a closer look then. The virus infects DNA again, so this is a second exposure. And that CRISPR complex is going to bind through homology because it has that same sequence. It took it from this virus. So it causes this bubble to form where the base pairs with the bottom because of that homologous base pairing that's from the viral DNA, that spacer sequence. And this Cas9 protein has 2 domains, a RUBC and HNH domain. They have DNase activity. So the RUBC will cut the top strand, and this HNH will cut the bottom strand. And what we're left with is blunt ends. So this whole viral sequence is now cut, and it can't do anything now, so it dies. So a question you might have is, why doesn't the bacterial genome, why doesn't that get spliced by this CRISPR system? Because this spacer sequence is technically a viral sequence. It's the exact match of a viral sequence. So there has to be something different. And if we look at the viral DNA, that protospacer, which we used, has a PAM sequence flanking it. So a PAM sequence typically is NGG, but different bacteria recognize different PAM sequences. And the Cas9 itself is on the lookout for this PAM sequence before it can activate and cut. So it has to see this. So even though we have homology to the bacterial spacer sequence, it doesn't have that PAM sequence. Typically it has a GTT or some other conserved short sequence that is never this PAM sequence. So and make sure that it's never that so that it never attacks itself. So if you're looking at this, it should be kind of obvious that we can use this in genome editing. So if we make a guide RNA, we just take that CRISPR RNA, the target sequence, which would typically just be part of that mRNA transcript that we did, which would be the spacer, and we would just attach it on directly to a tracer RNA and then add Cas9. And that's really all you need. And then you can focus on your target. You have to make sure it has PAM. Remember, this Cas9 complex is on the lookout for PAM. It's always looking for it. So you have to make sure that your target sequence here is always flanked by a PAM sequence. Now this isn't too hard because a lot of bacteria have different PAM sequences, so you can kind of find a specific Cas9 that fits your model. The general things we can do is we can take a normal gene, we could do gene replacement, or we just cut it out and put a new one in. We could do gene knockout, or we can do a gene addition. So CRISPR is already being used today. The USDA officially greenlit CRISPR in 2018. And so if we look at Penn State, they've actually engineered white button mushrooms to resist browning. So they took a oxidase gene, PPO gene, and then they had a CRISPR that was homologous to this target. They cut it out. And then this is called a gene knockout. And then now this mushroom is going to be mostly white and it will resist browning. So aside from making mushrooms white and resist browning, there's actually other pretty big implications of CRISPR in agriculture and other fields. So if we look at agriculture, a big problem with citrus is citrus greening disease. So if you look at Florida or California, it's a major problem. So they're utilizing CRISPR to create resistance. They've also used CRISPR with maize or corn to improve the yield during drought stress. And there's another company called Arvigenics using CRISPR on pennycress to increase that oil and meal quality so that when it's used as a cover crop, it can also be used to make profit. So it's profit and sustainable. This is being applied more and more into the medical field. So you can look at cystic fibrosis, single gene defects, and other diseases like Hunter syndrome, sickle cell anemia. And we can treat mosquitoes. So they've been using CRISPR to treat mosquitoes to make them bad vectors so that hopefully one day they can't spread plasmodium, which is malaria. So far I've only been talking about DNA viruses entering, and I utilize the Cas9 system. But what about RNA viruses? There are CRISPR systems that can deal with RNA, and one of these is the Cas13A protein, and this is the class 2. You can look more into that. But this protein is becoming more and more popular because of its uses experimentally. And so I'm going to go into this protein Cas13A and this system really quickly just to show the implications of this. So it doesn't have a DNA's binding domain because it's going to be attacking RNA. So it has a heparin domain, a higher eukaryotic and prokaryotic nucleotide binding domain. So it cleaves RNA. That's what this domain does. So we have Cas13A. So there's binding via a target sequence. So it finds that RNA virus, in this case, the RNA virus, and it does homology. And remember, it acquired that through spacer acquisition. So it took that RNA and then inserted it as a DNA into CRISPR locus or CRISPR array. And so we have this homology, and we don't have a PAM, but we have a PFS. It's very similar, but it's a bit easier to get because it's a 3' end, and it consists of a single A, U, or C base pair, unlike PAM, which is typically NGG, which is 3 nucleotides. So another big difference that Cas13a has when compared to Cas9 is that once it becomes active, it does not revert back to the inactive state. So once it has that homology base pairing and we have a conformational change to make it activate it, it stays active. And then it just starts cutting all RNA around it. And we think that happens probably because it starts to trigger apoptosis in the bacteria. So it's saying, hey, you're infected now. Let's just take you out and prevent this virus from getting anywhere else. And so this protein becomes almost like a monster protein. It just starts to destroy any and all RNA around it. And so you would have all this RNA coming through, and this protein will just destroy it all. And this is going to be called nonspecific RNA degradation. How can we utilize this? Well, they've been utilizing it as a test on called Sherlock. So you take RNA, you're going to amplify that RNA, make a large amount of it. You add Cas13a, and then once it has that homology, it goes crazy and it cuts all the RNA around it, including a reporter RNA, which produces fluorescence. So just because this is interesting, I'm going to focus a little bit more on this test and break it down because it's kind of a good thing to know. So this is the original sample. And in the sample, we might have the RNA that we're targeting. So let's say we're targeting Zika virus or something like that. We need this viral sequence for homology, but it's bound up in the genome. So we need to isolate the RNA from the rest of the genome, and then we need to amplify it. And so they do something called RT-RPA, which is Recombinase Polymerase Amplification. Much like PCR, RPA amplifies its sequence of interest. But where it differs is that PCR is using thermal cycling. So this is why it's kind of expensive. You're separating the strands by introducing heat, 95 Celsius, break it apart, and then you're going to start sequencing it to amplify that DNA sequence that you're interested in. Isothermal is more involved when we're talking about low budget or on the site kind of detection. So we need a constant temperature. So about 25 to 42 Celsius is where RPA functions at. And how it gets away with this is that it uses these proteins called the recombinase, and then it uses single-stranded binding protein, and then it's a DNA polymerase, and then 2 different primers. And so what it does is we have this recombinase protein that binds to oligonucleotides. This is what we feed it. These are the primers. And to make them target a homologous sequence. And so they will bind because of that homology from that nucleotide we provide it. And they will bind because that recombinant protein is going to cause a bubble and allow those primers to go there. Single-stranded binding proteins help stabilize this bubble, and then DNA polymerase can amplify this. And so now we end up with something that looks like this. This isn't really what we wanted. We only wanted this to this sequence, but it keeps going on. So we do this again. So we do another round of cycling, and we would end up with something like this for one of the strands. And if we do another round, we finally get our sequence that we wanted, that specific RNA sequence. And so this is an exponential growth where we would get a lot of these guys after a couple of rounds. And so now we have amplified our target DNA. We could go into where this Cas13a is utilized. So we have our amplified RNA, and we add Cas13a with our guide. So now there's this base pairing, and then we add a quenching reporter pair RNA. So what's interesting about this is that this quenching region is actually inhibiting the reporter from fluorescing. As long as this mRNA is intact, this is not going to fluoresce. But when it's cut, it will fluoresce, and we will be able to visibly see this. So remember that once that Cas13a is activated, it stays active and just starts cutting all RNA around it. So we would get collateral cleavage, releasing this fluorescent, and then we can visibly see that there's viral RNA present. So we'd be able to say, hey, look, there's Zika here, or there's dengue, or whatever you're looking at, whatever target you amplified. So let's just do a quick summary. So there's acquisition step for all the CRISPR systems where a sequence is put in as a spacer, like these guys, and then it's flanked by these repeat sequences. So if we break it apart and look at the Cas9 versus the Cas13a, you can see that this is looking for PAM. We're looking at the Cas9, and the Cas13a is looking for a PFS region. And the biggest difference are the domains. So this has a heparin domain to cut RNA, whereas the Cas9 has a rough C and H and H domain to cut DNA. Cas9 reverts back to the inactive form, and then Cas13a stays in the active form. So because of that, Cas9 is good for genome editing, and Cas13a is good for detection assays, like I've already described. So if you guys are interested in more, these are some of the links of where I got a lot of this information from. Feel free to explore more into both of these systems because they're pretty interesting.